Hi students, welcome to Ligis Academy. In this video, we are discussing about transcription stages in prokaryote. Transcription in prokaryote occurs within cytoplasm. Transcription contains three stages. One is initiation, then elongation, finally termination. The initiation stage consists of 4 steps: promoter recognition, promoter opening, abortive initiation, at last promoter escape. We will discuss details about these stages in initiation. Initiation is the first stage in transcription. Initiation describes the synthesis of first nucleotide bonds in RNA. In this stage, the enzyme remains at the promoter. The initiation phase is protracted by the occurrence of abortive events, in which the enzyme makes short transcript, releases them, and then starts synthesis of RNA again. The initiation phase ends when the enzyme succeeds in extending the chain and then leaves the promoter. The initiation stage consists of 4 steps. First one, promoter recognition. Second, promoter opening. Third, abortive initiation. And final one, promoter escape. First step, promoter recognition. In this stage, the recognition of the promoter by the sigma factor. The holo enzyme consists of the initiation factor sigma that interact with the sequences in promoter. We already discussed about the structure of sigma factor in earlier video. In that, the sigma has 4 domains and with each has conserved regions. Here the sigma 4.2, that means the second conserved region within the 4th domain, that interact with the sequences in -35 element, and the sigma 2.4 that bind with the sequences in -10 element. Sigma 1.1 block the entry of duplex DNA into active site. Sigma 1.1 act as a auto-inhibitory domain that block the binding of free sigma factor into the promoter. It releases the sigma 1.1 when it binds with the holo enzyme. When sigma factor bind with the core enzyme, a conformational changes occur and it releases the sigma 1.1 activity. When sigma 3.2 loops project into active site of the RNA polymerase, the DNA remains in a duplex stage. That stage is known as closed promoter complex. During this stage, the DNA remains in duplex stage. That is, no melting of the DNA occur. This temporary stage is known as closed promoter complex. Next step, promoter opening. Promoter opening, in which the sigma factor 2.3 contains the amino acid sequence, that is hydrophobic patch of amino acid sequence, phenylalanine, tyrosine, tryptophan. This hydrophobic patch flip out the first adenine in -10 element. This will create a flip, and due to the disruption of base pairs and weaker hydrogen bonding in AT region, the torsional strain will melt the DNA, and this melting of the DNA will leads to the formation of transcription bubble, and this conformational changes will exit sigma 1.1 from the opening of active site. It leads to the formation of open promoter complex, and melting extends beyond the transcription start site, and the RNA polymerase active site, that is beta-beta dash pinces, clamps on the downstream double-stranded DNA. Through the secondary channel, ribonucleotide enter the active site with a bound magnesium. This picture represents the structure of holoenzyme and the domains of sigma factor. The sigma 4.2 that in blue color interact with -35 element and the sigma 2.4 that interact with the -10 element. There is a sigma 1.1 that acts as an auto-inhibitory domain, also indicated here in purple color. Then comes to this third step, that is abortive initiation. In this stage, the polymerization of ribonucleotide begins. That is, the first ribonucleotide added to the start site, then the following the RNA synthesis occurs. Size of the RNA-DNA hybrid increases. The RNA polymerase do not move during this stage, as they are anchored to -35 element. And the synthesis of RNA-DNA hybrid without RNA polymerase movement explained by this scrunching model. Sigma factor is no longer necessary for this stage. As the RNA polymerase synthesizes RNA-DNA hybrid, it pulls DNA onto to itself. So during this pull, the potential energy stored in unscrunched DNA released upon unscrunching, and it facilitates the promoter escape. During abortive initiation step, the RNA synthesis takes place up to 9 base pair. If RNA-DNA hybrid is around 9 base pair during unscrunching, they clash with the sigma 3.2 loop. So melting of the RNA-DNA hybrid occurs. The initiation is aborted. That is, the short transcript about 9 base pair releases through this secondary channel and the initiation restarted. Then the final step in initiation is the promoter escape. In this step, the initiation complexes escape from promoter. When RNA-DNA hybrid size more than 12 base pairs, the unscrunching will displace the sigma 3.2 loop from active site. It destabilizes interactions between sigma 4.2 and -35 and between sigma and beta subunit. So sigma may or may not release from the core promoter, but it destabilizes the interactions between the sigma and -35. So RNA polymerase able to synthesize RNA-DNA hybrid beyond 20 base pair. This is known as the promoter clearance. Due to destabilization of sigma 4.2 and -35 element interactions, RNA polymerase is now free to move from promoter. So this free to move of RNA polymerase from promoter leads to the promoter escape. Several rounds of abortive initiation leads to promoter escape. After initiation, this initiation complex transforms into elongation complex. Then that's all about the initiation of the transcription. Initiation consists of 4 steps: promoter recognition, promoter opening, abortive initiation, promoter escape. After promoter escape, it leads to the next stage, elongation. First step, promoter recognition, in which the sigma factor recognizes the promoter element. Then promoter opening, in which the -10 element melt and the formation of transcription bubble occurs in this stage. Next stage, abortive initiation, where the ribonucleotide polymerization begin, but due to scrunching and unscrunching of the RNA-DNA hybrid, short RNA transcript releases this through this stage. Then the final stage, promoter escape, where RNA-DNA hybrid is more than 20 base pair. It destabilizes the interactions between sigma factor and the promoter element. So the RNA polymerase is free to move. It will lead to the promoter escape. Then leads to the next stage, elongation. Next step, elongation. During elongation, enzyme, that is RNA polymerase, moves along DNA and extend the RNA chain. The nucleotides are covalently added to its 3' end of growing chain. It occurs with the help of transcription elongation complex. This transcription elongation complex consists of core polymerase, and it also contains transcription elongation factor instead of that initiation factor in initiation. Transcription elongation complex is shorter than the initiation complex, but it has high affinity. This picture represents transcription elongation as well as the movement of RNA polymerase. Then discuss about the structure of transcription elongation complex. Transcription elongation complex consists of several sites: site of duplex DNA entry, unwinding point, non-template channel, RNA-DNA hybrid formation site, RNA-DNA hybrid melting site, template DNA exit channel, rewinding point, an RNA exit channel, and secondary channel. Then comes to mechanism of translocation. DNA template and RNA-DNA hybrid movement through RNA polymerase is refers to as translocation. Energy required for translocation is derived from pyrophosphate hydrolysis and thermal energy in the system. The mechanism of translocation is called Brownian ratchet. It consists of two poles, stationary pole and reciprocal pole. Stationary pole prevent the backtracking of RNA polymerases, and reciprocal pole allow the RNA polymerase translocate along template. And it consists of RNA polymerase consists of bridge helix and trigger loop, and the trigger loop has two conformations. In the presence of NTP, it has conformation changes to ordered conformation, and in the absence of NTP, disordered open conformation. Steps in translocation. In post-translocation stage, RNA polymerase have a new template base exposed in active site. This ribonucleotide hybridized with the template and blocking the backtracking of RNA polymerase. Trigger loop then forms a closed conformation when RNTP ribonucleotide binds with it. So it forms a ordered conformation. So enzyme is in the pre-translocated state. When the ribonucleotide form a phosphodiester bond, it induces a conformation change in trigger loop, and this conformation change leads to the disordered open conformation in trigger loop. This trigger loop has an intermediate state with elongated conformation. It presses against the bridge helix that pulls RNA-DNA hybrid. So the enzyme translocated. Then these steps repeated. Then talk about elongation factors. There are three main elongation factors discussed here: NAS-G, NAS-A, Gray proteins. NAS-G means N-utilization site protein G, NAS-A N-utilization site protein A, and Gray protein A and B. First one, NAS-G or N-utilization site protein G. It increases the processivity of RNA polymerase. They function as a clamp and bind downstream of duplex DNA. It prevents the posing of RNA polymerase. So it promotes the row dependent termination also. Then NAS-A. NAS-A stabilizes RNA duplex, which increases the RNA polymerase posing. So it decreases the efficiency of termination. NAS-G increases the processivity of RNA polymerase. Then NAS-A decreases the efficiency of termination. Then Gray proteins, that is Gray A and Gray B proteins. Transcription is not a continuous process because RNA polymerase briefly pause due to availability of ribonucleotide, errors in template, formation of secondary structures, etc. So most posing are transient, that elongation will restart immediately. But in some cases, post RNA polymerase can backtrack and creating a RNA flap that can extend into the secondary channel of the RNA polymerase. Gray proteins have a catalytic domain that inserted into the secondary channel of RNA polymerase. In the case of Gray protein A, induce the hydrolysis of short oligonucleotide up to 2 to 8 nucleotide, and they prevent the formation of arrested transcription elongation complex. Whereas the Gray protein B induce hydrolysis of large transcript, that is more than 18 nucleotide, and they rescued the arrested transcription elongation complex. Then last stage of transcription, termination. Termination involves recognition of point at which no further bases should be added to chain. During elongation, the RNA chain length increases. When the RNA polymerase reaches terminator sequence, the termination occurs. When last base is added to RNA chain, transcription bubble collapse. Then RNA-DNA hybrid disrupted. Then the DNA reforms in duplex stage. Then the enzyme and DNA are released. This is the final stage. Termination include two pathways. One is row independent termination, otherwise known as intrinsic termination, and the other one is row dependent termination. Intrinsic termination or row independent termination. As the name indicates, there is no row factor. Majority of the bacterial termination is row independent. Intrinsic terminator located within the RNA. That is, the termination signals comes within the RNA itself. The features of RNA must contain a GC-rich inverted repeat that forms a stable stem loop structure, then followed by a short stretch of 8 to 10 uridine nucleotides immediately downstream from the stem loop. Then we discuss about the mechanism of intrinsic termination. The RNA polymerase briefly pause when a stem loop structure formed behind. First, there is a stem loop structure formed in RNA due to its GC-rich region. RNA polymerase briefly pause when stem loop structure formed behind. Then the following adenine-urine-rich RNA-DNA hybrid inside the active site of RNA polymerase have a weaker hydrogen bonding. So this RNA-DNA hybrid will peel off from this transcription elongation complex, and the transcription elongation complex disassemble. It leads to the termination of the transcription. In the case of intrinsic termination, the sequence specificity of stem loop is irrelevant, but where the deletion of uridine will abolish the terminator activity. Then comes to rho-dependent termination. As the term indicates, the termination occurs with the help of rho factor. What is rho factor? Rho factor is a RNA-DNA hybrid helicase enzyme. It is hexameric in nature, and each subunit have two RNA binding sites. One primary binding site located on N-terminal and secondary binding site located on C-terminal. Then comes to the term rho factor, rho utilization site or RUT site. RUT site is a specific site on which rho factor recruited. It is a cytosine-rich site. These are the steps involved in rho-dependent termination. When during termination, the rho factor binds to the RUT site. That is, rho factor binds to the cytosine-rich RUT site. This RUT and rho hybrid pulls the RNA in 5'3' direction. They eventually catch up with a post-RNA polymerase in front. And this rho unwind RNA-DNA hybrid and leading to termination. That is, during termination, rho factor binds to the right side and the rho factor pulls the RNA in 5'3' direction and unwind RNA-DNA hybrid, and this will lead to the termination. In this process, that is rho-dependent termination, termination is regulated by a protein called NUSG, N-utilization site protein G. This NUSG protein interact with the rho factor and recruit rho factor into RUT site. The same domain in NUSG also interact with the NUSE, which bind with the ribosome. Normally, NUSG interact with ribosome because in case of prokaryote, transcription and translation occur simultaneously. So, in case of prokaryotic, transcription and translation occurs simultaneously. When reaching stop codon, ribosome releases NUSG protein. This NUSG protein recruits rho factor into RUT site. It leads to the termination. So, in the case of prokaryotic transcription, complete translation of mRNA available before transcription termination. So, there is any error occurred during translation, ribosome stops and it recruit NUSG. Then this NUSG recruit rho factor into RUT site. This leads to the termination of transcription. So, any error in translation will stall the ribosome, which enable NUSG recruit rho and terminate the transcription of mRNA immediately. Hope this video useful to you. If you like this video, Please share, like and subscribe. Ligase Academy, your educational catalyst. Stay tuned to next video.